here in attendance, as well as the other speakers. My name is Alyssa Griffin. I'm an assistant professor at University of California in Davis, and I'm in the Department of Earth and Planetary Sciences, as well as an appointment at the Bodega Marine Laboratory in Bodega Bay, California. So today, I'm excited to share with you some of the work my research group is doing at the intersection of carbonate sediments, carbon cycling and storage, and also coastal ecosystem resilience and change. So our research group at Davis uses marine biogeochemistry really as a tool to understand biological, geological, and chemical interactions occurring in our oceans over various spatial and temporal scales. And I think we can all agree that understanding marine biogeochemical processes is increasingly important as human-induced alterations across these systems and across our planet really continue to accelerate. Of those human-induced changes, one of the most important ones, I would argue, and I think all of us would be climate change. And climate change at this point has become so severe that current models show that in order to avoid the most severe consequences of the climate crisis, we must reach net negative emissions, which happens only when we're removing more carbon dioxide from the atmosphere than we're putting into it. And to reach net negative emissions, this will require, first and foremost, and I want to make this abundantly clear, large-scale and rapid emission reductions, which are shown in this plot in brown. But unfortunately, we've backed ourselves so far into a corner that I think we have to face a pretty harsh truth that we'll have to develop carbon removal technologies at the gigaton scale within the next decade, which is shown here in blue. And this need to reach net negative emissions and develop these technologies has really led people to look to the ocean for solutions. And this makes a lot of sense since the ocean is the largest active carbon reservoir on the planet. And it's already estimated that without any intervention, that 30% of fossil fuels emitted from land use changes and fossil fuel emissions, 30% of those are being taken up by the ocean annually. And once that ocean is in the carbon, it can, once that carbon is in the ocean, it can react in a variety of ways, but ultimately is sequestered in marine sediments on the seafloor. So the previous talks really teed me up here, and I thank them for that. So our group focuses a lot on marine sediments and sedimentation because they really are the ultimate sink of atmospheric carbon dioxide over geologic timescales. And therefore, marine sedimentation and sedimentary processes are fundamental controls on ocean chemistry, atmospheric CO2 levels, and Earth's climate through time. But one thing I'd like to emphasize in this figure is that all of these feedback mechanisms of the long-term carbon cycle that are pictured here, including marine sedimentation, play out over millions and millions of years, which is far too slow to help us reach that gigaton removal target in just a few decades. And so when folks talk about marine carbon dioxide removal, really all of these technologies are either aiming to increase the rate of oceanic uptake of atmospheric CO2 into our oceans, or increase the rate of sedimentation, the sinking and storage of carbon. And ideally, a technology would be able to accomplish both. But what I think is important to remember is that all of this change, human-induced climate change, as well as many of these proposed climate interventions, are situated within the coastal ocean, where carbon dynamics are governed by a multitude of drivers, many pictured here, including watershed processes, ecosystem metabolism, a whole suite of physical processes as well. And because coastal carbon dynamics are so complex, it can be really difficult to track the sources and sinks of carbon within the coastal ocean. And in turn, this really inhibits our ability to predict how these individual processes and coastal oceans and ecosystems will respond to future climatic changes or interventions. And predicting these changes has important implications for not only local carbon dynamics within the coastal ocean and coastal ecosystems, but also global carbon dynamics, because the coastal ocean acts as this really important link between the terrestrial and oceanic carbon cycles. In this picture, I would argue that perhaps one of the most understudied sources and sinks or processing of carbon in this environment is sedimentary exchange. And so you can see that it's highlighted here in this diagram as having an important role in coastal carbon dynamics. And yet we really don't have a lot of good understanding, especially in the very near shore environment, of what these sedimentary exchanges look like across the sediment water interface. And so a lot of my talk today is going to be focused on this arrow that I've highlighted here. And trying to better understand those fluxes has led us to our sort of broad research questions in our group that I'll be discussing today, which are, what role do marine sediments play in the cycling of carbon in coastal ecosystems? And ultimately, can we leverage the natural processing, storage, and fluxes of sedimentary carbon to mitigate future climate scenarios? And I'll be addressing these research questions in two nearshore environments. So we've already talked a lot about coral reefs today, but I'll also be discussing some carbonate sedimentary dynamics in seagrass environments as well. But we'll go ahead and start in coral reefs. So again, the previous speakers have really made the setup of my talk much easier, but coral reefs have received a lot of attention in the context of global climate change and oceanic change, not only because of their ecological importance and the high levels of biodiversity that they support, but because they're made almost entirely of calcium carbonate. So when we think of things like ocean acidification, coral reefs are really threatened on every front. And because reefs are made entirely of calcium carbonate, we can actually think of them as a balance or a budget of carbonate material. And so this balance is often referred to as net community calcification, which has been discussed already today. But just as a reminder, net community calcification, or NCC, is the balance of the production of calcium carbonate and its destruction. And some of these productive processes include biological calcification, but also inorganic precipitation. And the destructive processes really hinge on gross dissolution. And so just like any budget, if we're making more of something, if we're putting more into the system than we're taking out, we can look at the reef as it being in a state of net growth. And so overall, our reef is growing and our net community calcification is positive. But if you are taking out more than you are putting in, then we can anticipate reefs transitioning into a state of net loss. And current predictions of oceanic change suggest that reefs around the world will transition to this state of net loss or negative net community calcification by the end of the century. And so some solutions have been proposed to address this loss, and one of them is ocean alkalinity enhancement. So if you're not familiar with ocean alkalinity enhancement, it's essentially the idea of adding alkaline materials in either solid or solution form to marine environments. And through this addition of alkalinity, we're not only decreasing or mitigating acidification, but we also increase the ocean's buffering capacity, which means the ocean now has a greater ability to take up carbon dioxide. And this approach has been proposed, again, to reverse acidification impacts on reefs. But many of these studies are model-based and haven't been performed in situ. And actually, the only in situ alkalinization experiment I'm aware of, I'd love to hear if folks have heard of others, is this one by Rebecca Albright et al., which came out in Nature several years ago now, where they alkalinized a coral reef ecosystem and found that it increased net community calcification by about 6.9%. And so this was really exciting, promising, but one thing that this study couldn't get at was that increase in net community calcification due to increase in production or decreases in destruction, or some combination of both. And this is important to think about in a coral reef ecosystem, because all of these destructive and productive processes are happening not just in the coral reefs and the reef framework, but also in the surrounding sediments. And so carbonate sediments, in fact, are often the largest reservoir of carbonate in this overall reef budget. And so if we want to understand how a reef ecosystem as a whole is responding to an intervention like alkalinization, we really need to understand how these individual components of the system are responding, and then bring it together to understand the ecosystem scale response. And so I wanted to answer the question of how are reef sediments responding to alkalinization. So to conduct this work, we wanted to investigate these dynamics in shallow reef sediments. So one thing that I don't think's been highlighted yet today is that in shallow carbonate sediments, we know that there are these diel cycles of photosynthesis and respiration happening. And those cycles of photosynthesis and respiration will take up and release CO2 respectively. And these are occurring in the sediments over daily cycles. And in turn, if we look at the equation that we've seen multiple times already, those daily changes in sedimentary CO2 levels are responsible for then driving cycles in carbonate precipitation and dissolution as well. And typically what we see is that during the day we have net photosynthesis, which is drawing down CO2, increasing your carbonate saturation state, and leading to carbonate precipitation. And then the opposite at night, where you have elevated CO2 from net respiration driving net carbonate dissolution. And these dynamics, we can get at them by measuring things like dissolved inorganic carbon, which is just the total amount of inorganic carbon dissolved in seawater, as well as total alkalinity, which is a solution's acid neutralizing capacity and is largely related to the amount of negatively charged ions in seawater, the primary ones I've listed here in this equation. And you can see the carbonate system is a big component of that. But these daily cycles and changes in dissolved inorganic carbon and total alkalinity due to these coupled effects of photosynthesis and respiration and carbonate dissolution and precipitation can actually lead to chemical gradients between the sediments and the overlying seawater. And this results in fluxes of both dissolved inorganic carbon and total alkalinity across the sediment water interface. And these fluxes are really useful because in measuring them, we can learn more about the biogeochemical processes occurring within the sediments themselves. So to answer this question about alkalinization in reef sediments, we also worked in Lizard Island. It's just, such an awful place to work. That's why everyone goes there. But we looked at 4 reef environments, so contrasting sedimentary environments around the southern shores of Lizard Island. And to measure the fluxes across the sediment water interface, we used sediment incubation chambers. I know some folks on the call are familiar with these, but in case you aren't, I have a little video here. But essentially, that's me on scuba deploying these chambers, and we use them to measure these fluxes, again, across that interface. These are really useful because we can sample through the sampling port using these syringes and then take them back to the laboratory to analyze them for the parameters that we're interested in. But what's also useful about that sampling port is that we can actually inject different treatments into the overlying water within the chamber to achieve differences in water column chemistry that we might want to mimic. And so in these experiments, what we did is we injected acidified water and alkalinized water into the chambers and then measured the fluxes over a 24-hour cycle. And those spinning acrylic discs are there to promote advection because these are advective sediments and we're trying to mimic the natural physics of the environment as much as possible. So using these chambers, we measured net community calcification, which I already mentioned is that balance of carbonate production and destruction. And we also measured net community production, which is the balance of gross primary production and respiration. We ran these chambers for 24 hours and we sampled them at dusk and dawn to capture the nighttime rates in these parameters, and then again at the following dusk to capture the daytime rates. And by injecting that high alkalinity water and that acidified water, we were able to capture a wide range in the overlying saturation state within the chambers. And so I'm going to show you some data, but this plot is a little dense, so I'm going to walk you through it first. So on the left side here, we have the net community production rates. On the right side, we have the net community calcification rates. The data are colored by the experimental treatment with acidified conditions in red, the controls in green, and the alkalinized in purple. And the somewhat faded colors here, I don't know if that's showing up on folks' screens, but the somewhat faded colors represent our nighttime rates. And then the solid colors represent our daytime rates. And here in the bottom panel are our rates integrated over 24 hours. And so if we first look at the net community production rates, what we see is that there was no significant difference at any of our sites in net community production across our different treatments. And so this suggests that photosynthesis and respiration are really not influenced by at least the changes that we induced in carbonate chemistry in these sediments. And this is really in stark contrast to what we see in the net community calcification rates. So I'm going to start with the nighttime rates here. I know I have a little sun there, sorry. It's supposed to be a moon. But our nighttime net community calcification rates did differ across treatments as we would expect, with lower net community calcification happening under acidified treatments relative to the control, and higher NCC rates in the alkalinized treatments relative to the control. What was interesting, though, is that we actually didn't see a difference between acidified and control treatments in the daytime net community calcification rates. Even though net community calcification was increased under alkalinized conditions, again, the acidified and control daytime NCC rates showed no significant difference. And I think this supports prior evidence that there's some kind of biological activity that's buffering the system during the day against that acidification, whereas at night that activity is not occurring, and so it's really just purely geochemical dissolution controlling the system at night. What's interesting, though, is that over 24 hours, we see that across the different treatments, that the acidified treatments fell below this threshold of net dissolving. And so if you see this dashed line here above it, the sediments are net precipitating or net producing carbonate. And below that line, we're losing carbonate from the sediments. And because there was no difference between acidified and control treatments during the day, this result in our 24-hour experiment suggests that increases in nighttime dissolution rates are really what's driving differences in acidified and control conditions over 24 hours. So kind of an interesting find there. If we look at how alkalinization influenced the sites, all of the sites were net precipitating during day and night under alkalinization, as well as over 24 hours. And we saw that alkalinization increased net community calcification by 158% per unit increase in saturation state. And interestingly enough, that 158% per unit omega is really similar to this global study. I think Olivier showed this earlier from Brad et al., where they were showing only acidified conditions. And so this suggests that we can extrapolate this relationship to higher saturation states as well. When we think about this in the context of MCDR or MCDR efficiency, we did see an increased uptake in DIC, which suggests that there is some MCDR capacity for this approach. These are phase diagrams with total alkalinity on the y-axis and dissolved inorganic carbon on the x. And the colored lines represent PCO2. And the triangles here are showing our control conditions, and the squares are showing our alkalinized conditions. And without getting too much into it, you can just trust me or ask me later. This bar here represents that total amount of additional uptake that we can see in the sediments. And that ranged from about 75 to 200 micromoles, which equates to a OAE uptake ratio or a CO2 uptake ratio of about 0.36 to 0.54 across all of our sites. And what's interesting is that this MCDR efficiency is somewhat lower than what we've seen in other systems because of that increased calcification. And so it kind of highlights these potential trade-offs of promoting ecological benefits while also maximizing potential carbon storage through some of these interventions. All right, so moving into my second favorite nearshore environment, looking here at seagrasses. So I'm going to move a little bit into organic carbon, but I swear we'll bring it back around to carbonates. So we know that the coastal ocean is a hotspot for sedimentary organic carbon storage, as shown here in this global heat map of seafloor carbon storage. But what we also know is that some ecosystems are even better at storing organic carbon, and we refer to these as coastal blue carbon ecosystems. Blue carbon is kind of like this catch-all term for any carbon that's captured in the world's oceans and ecosystems. But coastal blue carbon is really highlighting these vegetated ecosystems that store a disproportionate amount of carbon in both their biomass and their underlying soils and sediments. Just to give you an idea of how good they are at doing this, mangroves and tidal marshes remove carbon from the atmosphere at about 10 times the rate than tropical forests and store potentially 3 to 5 times more carbon per acre. And seagrasses occupy only 0.1% of the world's seafloor, but it's been estimated that about 11% of the ocean's buried carbon resides in seagrass sediments. And coastal blue carbon systems are distributed around the world. They're on the coastlines of every continent except for Antarctica. And they provide a whole suite of co-benefits in addition to carbon storage, such as water filtration, habitat, tourism, shoreline protection, etc. But like most things, humans don't know what they have, and we are destroying these systems at pretty alarming rates. And when we destroy these systems, we're potentially releasing some of that stored CO2 back into the atmosphere. And when we look at seagrass carbon dynamics, they're really complicated because seagrasses have this really broad latitudinal range. So that means they can grow in a variety of different climates and sedimentary environments with varying physical and geochemical properties. They also interact with the sediments in a variety of ways, including potentially increasing particulate deposition and subsequent burial. They can reduce resuspension by stabilizing underlying sediments. They introduce oxygen into the sediments through their roots and rhizome systems. And they've even been shown to enhance microbial activity within the sediments. And all of these processes and properties together can significantly influence the chemical cycling of carbon within the sediments, and in turn, the exchange of carbon and alkalinity across the sediment-water interface. But when we think about blue carbon systems, I would say traditionally we've really only thought about them in the context of this canonical carbon, organic carbon burial and storage. And so some of the work we do does try to quantify that organic carbon storage. So this is just some of our methods. We take these sedimentary cores, we acidify them to see how much inorganic carbonate is present, and then we also measure the organic matter as well as associated parameters like grain size, C to N ratios, and associated isotopes. But when we do this work, what we see is that even at a meadow scale, we see tremendous variability in this organic carbon storage. So this is a paper by Dr. Lena Capiz and myself and others that just demonstrated that intrameadow variability of organic carbon storage can actually be greater than regional variability. But what we don't know is what's driving this variability. What are the mechanistic drivers of these really huge differences that we see across and within systems? And so I'm going to talk for a second about the chemical cycling here and what that means for the fluxes across the sediment-water interface. And as Olivia was talking about earlier, the longer you spend time in sediments, the more you come to understand that it's all microbial. It's all about the microbes. And in sediments, we have these different redox zones that facilitate different organic matter remineralization pathways by different groups of different microbial groups. And so ultimately, microbes are largely determining what carbon is sequestered in the sediments and what is returned to the ocean and ultimately the atmosphere. And so we had the question of, can we get at what role do microbial communities play in carbon storage variability that we see at this meadow as well as this regional scale? And so we went back and we resampled sites from Lena's work, and we quantified the geochemistry, the carbon, but then we also aimed to characterize the microbial sediments within these sediments as well, and identify their abundance as well as what they're doing. And this is very preliminary work, so I'm just going to share via text some of the main findings, but what we do see is that there are significant differences in the microbial communities between high and low carbon samples within a given meadow, and that these differences in microbial communities are largely explained by the presence of sulfur cycling microbes, so microbes that are associated with either sulfate reduction or oxidation. And through some of our other geochemical work, we saw that these higher carbon samples were also associated with higher inorganic carbon content, as well as lower delta 34S sulfur isotope values. And our working hypothesis now, as I said, stay tuned, is that potentially sulfate reduction in these systems is leading to increased recalcitrance of the organic carbon that's present, and also potentially driving carbonate precipitation in these systems. And again, this matters. I said I bring it back to carbonate dynamics because again, this can heavily influence these fluxes of dissolved inorganic carbon and total alkalinity. So now we're looking at a more complex picture where not only do we have photosynthesis and respiration, we also have calcium carbonate dissolution and precipitation, but also these redox dynamics that are playing out in different environments. And these fluxes of dissolved inorganic carbon and total alkalinity are not inconsequential. It's been suggested that these exchanges across coastal sediment interfaces could be laterally exported into other environments, and that this represents potentially a carbon sink that's 4 or maybe more times greater than this traditional organic blue carbon sink that we typically associate with these sediments. And so some work we're doing right now is looking at these benthic alkalinity fluxes in the estuarine system in Northern California. This is a long-term data series here where we see this source of alkalinity in the Back Bay here, where we have all of these vegetated benthic environments like seagrasses and salt marsh habitats. And so we're working in this relatively pristine system of Tomales Bay, which is also well studied hydrographically. It also supports a lot of commercial aquaculture to see if we can't identify, if benthic fluxes are indeed the source that we're seeing in the Back Bay. Olivia mentioned this modeling study that was done a few years ago. We're also looking to measure some of these outcomes from the model in situ. So we're looking at carbon sulfur alkalinity dynamics in seagrass environments, not only in our backyard in temperate siliciclastic sediments, but also in carbonate-rich organic matter poor sediments. So thinking about like seagrasses that are adjacent potentially to coral reef systems. Because these dynamics are going to look very different because of differences in organic matter and iron. And some of our preliminary data there, I know I'm getting low on time, so I'm going to hustle through these last few slides. But one thing I didn't mention in the previous work is that we actually measured alkalinity fluxes from a beach sand and a beach seagrass at the same site. So in and outside of vegetated sediments. And what's interesting is that we saw significant differences in the alkalinity fluxes again at night. So greater dissolution at night in the seagrass system, but also higher net precipitation during the day. And so actually, when we integrate that over 24 hours, we really didn't see a difference between net community calcification in the bare sediments versus the vegetated sediments. We're also doing this work, like I mentioned, in our local temperate environments. So this is my graduate student, Liu McConan, who's working on quantifying pyridization and what that means for alkalinity fluxes in these systems. And so, yeah, just a few takeaways. I hope folks now see, if they didn't before, that carbonate dissolution and precipitation dynamics in coastal sediments are really significant in the context of coral reef's response to climate change as well as to climate interventions. And also thinking about some of these accounted for blue carbon sinks in seagrasses and other vegetated systems. Just acknowledging our funding sources as well as many, many students who have helped with this work and my graduate students who are pictured here. And yeah, looking forward to discussion and questions.